All right, we're at the top of the hour. I may just pause to let some people in, but today we have Th1nk with Dr. Ernesto Nakayasu. He's coming to us from PNNL. And a little bit about him. He received his degree in biological sciences from the University of Sao Paulo in Brazil and PhD in pathobiology from University of Texas, El Paso. After postdoctoral training at the Pacific Northwest National Laboratory, aka PNNL, and appeared as a scientist at Purdue, Ernesto returned to PNNL in 2015, where he's currently a senior scientist and leader of the Multi-Scale System Science Group. His research has been focused on the application of advanced mass spectrometry technologies such as proteomics, lipidomics, metabolomics, and analysis of protein post-translational modifications to study complex biological systems. In diabetes, Ernesto has been studying biomarkers predictive of type 1 diabetes development, molecular mapping of the pancreas, and mechanisms of beta cell dysfunction and death. I'm going to drop a couple of his recent papers that were well received. He wrote along with others in February 21 in bioarchives. They had a preprint regulation of beta cell death by ADP ribosyl hydrolase ARH3 via lipid signaling in insulitis. And then in January 31st, they also had a bioarchives preprint reduction of chemokine CXCL9 expression by omega-3 fatty acids via ADP ribosyl hydrolase ARH3 in Min6 insulin-producing cells. So I'll drop a couple of those in the chat for your perusal. And the title of his talk is The Yin and Yang of Fatty Acids in Regulating Beta Cell Inflammation. Thank you so much for joining us.
Monica, thank you so much for the invitation and for the opportunity to talk about our work. Here, as you mentioned, we're interested in all different kinds of molecules, and something that I've been working since my PhD is in lipids. And actually, when I finished my PhD, I kind of almost swear that I was never going to work with lipids anymore. But it always, coming back to me, so I gave up on saying that.
Well, they're pretty difficult to work with, as I recall.
Yeah. So, so today, I want to talk about lipids. Lipids. When we talk about lipids, the first thing comes to our mind is this like central function here. That's the cell membrane. But I'd like to remind that they have a lot of other functions like energy storage, insulation, and hormones. There are a lot of hormones that are lipids too. And but today I'll talk more about signaling lipids. If you think about the different signals in lipids, we have lipids that are more anti-inflammatory, like omega-3 fatty acids or these fatty acyl esters of hydroxy fatty acids, or what we call FAFAs. And in the other side, we also have lipids that are pro-inflammatory, like arachidonic acid, ceramide, free fatty acids. Those are so if you don't have a balance, you will shift to a state that will more be pro-inflammatory, and the cells will die. So our goal was to understand what is the role of lipids in apoptosis-inducing signaling beta cells. And so more specifically, like what are the functions of lipids in this process? So this is already published data, so I won't go into much detail here. But to understand the lipids, sometimes the lipid information we get from omics is a little bit, let's say, noisy. So what we did was to look at multiple systems and see what is really conserving signal. So those are probably we had a better chance to be something that worth to go after. So we use these endocrine beta H1 cells, and we treat it with cytokines. The same with human islets, and then also we got islets from NOD mice compared to normal mice, and then we did lipidomics of all these three models, and we found two very conserved signatures. One was that we could see phosphatidylcholines with polyunsaturated fatty acids on it, and lysophosphatidylcholines. And the other signature was the downregulation of triacylglycerols. So we went after the top one first, the PCs or phosphatidylcholines with polyunsaturated fatty acids in these size PCs. So when we took a closer look to these, we see that there were a lot of these polyunsaturated fatty acids, and then the relationship with lysopCs. So the lysopCs, they are formed by cleaving one of the fatty acids from the chain, and then they're forming these PCs. So others, so one enzyme that is known on the beta cell apoptosis is the calcium-independent phospholipase A2 or IPA2 beta over PRA2D6. So that signaling can release omega-6 fatty acid or arachidonic acid is one of those, and that trigger pro-inflammatory signaling in beta cells and triggers beta cell death. But I didn't want to go after this because there were a lot more things done already. The Ramana done and Mirma and other groups they have already shown work quite a bit on this side. So we went after the other side, which was these omega-3 fatty acids, and we were questioned that maybe this would have a protective effect, instead on the cells. And there was some evidence about this, in the first paper is from Marion Ruers that they showed that diet with omega-3 polyunsaturated fatty acids reduce the risk for you to develop autoimmune response. And the second paper is in OD mice that they show that if you feed the mice with omega-3 fatty acids, you reduce the onset of diabetes in these mice. And in both of these cases here, the reduction is about 60%. So this is a paper that we published last year, and Sam Stark already presented, so I'm not going to go in detail here because he already presented at this venue last year. But what we found that time was that the PCs of polyunsaturated fatty acids cleaved by the PRA2D6 and they release omega-3 fatty acids. And what it does, it leads to degradation of this protein called SUS12, which is a component of this histone methylation complex. So you have less methylation, and then that would release the expression of this protein ARH3, and that which is an anti-apoptotic and would lead to cell survival. So so the idea is that the ARH3 would reduce cytokine-mediated apoptosis and presumably by reduced ADP ribosylation. So I'll show a little bit more details about this. So ARH3 is here. Let me explain a little bit the pathway here. So for ADP ribosylation, you get this molecule, nicotinamide adenosyl dinucleotide, NAD or NAD, and then which are transferred by PARPs. And here, this schema is not complete, but there are 16 of those in the human genome, and they transfer these ADP ribose units to the target proteins and release nicotinamide. You can have just one unit, what we call mono ADP ribosylation, or you can have multiple units, which we call poly ADP ribosylation. And ARH3 is a molecule that is an enzyme that cuts off from the last unit from the protein, and more specifically hydroxyl groups from amino acids like serine. There are other enzymes like PARG would mainly cut like the poly chains, instead. So the idea that we have is that there are cytokines that induce some PARPs to make either mono or poly AD primabosylation, and then that will be part of the pro-apoptotic signaling. But then there might be other processes that being regulated by these PARPs as well, because there are 16 of those, and we don't know almost anything about this PARP in beta cells. So first thing we did was to look at the expression of PARPs in several models. So this panel A here is cells or human eyes were treated with different cytokine cocktails, and we see there are some that are and the expression are consistently regulated by the cytokine treatment, so like PARP 9, 10, 12, and 14. The bottom panel here, this is data from the HPAP. So these are single beta cell RNA-seq, and we can see here. So in these from non-diabetic donors, AAB1, AAB2 is whether they have one or two autoantibodies against the human islet proteins, and then T1D is after the onset. You can see the variety of these PARPs are also regulated by well, they are upregulated after the onset of the disease. So we selected based on these two panels here. We selected some of them to screen. We include PARP 1 and 2 because they're not regulated by expression themselves, and then we add these ones that are consistently regulated. And so then we screen for the ones that are capable, we screen for the ones that if we do an RNAi, if they would reduce apoptosis. So here CT stands for cytokine or non-cytokine. And then we can see that compared to the control, PARP2 is the one that is reduced when it's knocked down. So PARP2 is responsible to to trigger the apoptotic signal. We repeat this experiment a couple more times, so we were very confident on PARP2. But right now, what we're working on is to do proteomic analysis that we are enriching the peptides that are AD primabosylated to be able to determine what are the PARP2 substrates that is responsible for this signaling. But that's where we are on this side. We also went to look at other processes as well. So we did just proteomics. This is global proteomics without any enrichment. We did knockdown of ARH3, and we treated with cytokines, in this case, interferon gamma, IL-1 beta, and TNF alpha, and then we did proteomic analysis. So when when we look at the data, we see several different processes are being regulated, things like antigen presentation, necroptosis, or chemokine signaling. So we start to look more towards the ones that are related to the immune response. And then we look at the network of these immune response proteins. We see things in this side here are things that are mainly for antigen presentation. We see TAP2, so these are MBGC molecules, and here is the proteasome for processing. And this side here is more for the chemokine expression, and we were very interested in the chemokine CXCL9. CXCL9 is important for type 1 diabetes because it helps recruit the T cells to the site during insulitis. So that's why we were interested in CXCL9. And then if we look at ARH3, when we knock down ARH3, CXCL9 is increased with the cytokine treatment. We tested the different cytokines because we started with a cocktail. We didn't know which ones were responsible. but to make a long story short, here you need interferon gamma to produce CXCL9, but interferon gamma by itself is not enough. So interferon gamma by itself doesn't trigger the expression of CXCL9. You need to have a combination with at least interferon gamma or IL beta or TNF alpha. But with the trio, then you actually express more. Next, what we did, we went back to the omega-3, and we found that if you pretreat the cells with the omega-3 fatty acids, EPA or DHA, you significantly reduce the expression of CXCL9. We next we test if this was via ARH3, and then we can see here if you knock down ARH3, you actually increase the CXCL9 expression, showing that the response or the reduction of CXCL9 by omega-3 fatty acids is actually via ARH3. So the summary of this first part here is that we know now that PARP2 is responsible for the ADP ribosylation mediated apoptotic signaling. We're still working on that to really understand more of the signaling. ARH3 regulates a variety of processes in the cells, including antigen presentation, chemokine signaling, and then omega-3 fatty acids downregulate the expression of CXCL9 via ARH3 signaling. So now we're moving to the other side. Back to the 3 different lipidomics data. We went now to investigate this downregulation of triacylglycerol. One thing that we found was that there's this lipase called endothelial lipase or LipG. LipG is increased, the expression is increased in human islets treated with cytokines. And then at the same time that we see this palmitate being increased as well. So the idea is that this LipG is somehow cleaving these triacylglycerols and then releasing fatty acids like palmitate. So we went to look at some data from TEDdy because LipG is a lipase that cleaves plasma lipoprotein. So we say maybe there is some trends that we can also see in the human plasma. And in TEDdy, it has this very large study that they collect blood samples every 3 or 6 months all the way. I mean, the study that we looked was the nested case control study one that the endpoint was at 6 years old. And then we had a case of type 1 diabetes or the ones that just stay at autoimmune response at that time. And then we got, they collect a bunch of different data. So we request to look at their lipidomics data. And when we look at their lipidomics data, this is, we center the data based on the time that they have the autoimmune response starts. So they measure, they monitor for the appearance of autoantibodies. And then the way that when we start to see autoantibodies, we count as a seroconversion. And we can see just after seroconversion, there are several fatty acids start to increase in the plasma. And it's very temporal because after a while, it's all gone. At the same time, we see phosphatidylcholine and triacylglycerol being reduced. And also, I think this matches well with the LipG increase as well. Now we went to see if we can see some of these fatty acids free fatty acids in islets. So I contacted a collaborator of mine, Julia Laskin, and what they can do in their lab is what they call imaging mass spectrometry. So basically, they have this kind of apparatus that you can collect mass spectra from spots on the tissues. So you can move these throughout the tissue, and you can make images of molecules across the tissue. And if you look at those, we can see a bunch of fatty acids here. So, for example, Fa16-0 is palmitate, 18-0 is stearic acid, but there are some other peaks that are here are fatty acids as well. Here are some of the images. Some of these are some more complex lipids, such as phosphodioinositols or phosphodiesterins. You can see that some of them are enriched actually in these islets. You can see the islet regions here. But this bottom line here is all fatty acids. That we can actually see these free fatty acids on the islet itself. Then we went to and we asked the question if this free fatty acid might have a function in beta cells. In this case we use Th1nk cells, but when we treat with just the fatty acid themselves, they don't change much. They have some minimal effect, but when we combine them with cytokines, then they have a much different pattern. For example, here, palmitate, for example, with combined with cytokines, basically doubles the apoptosis compared to the cytokine. Despite like here being just palmitate is only like maybe 10% difference. EPA, for example, is another good example that basically abolished almost completely all the apoptosis here. Here's just those response curve. With just palmitate, there is just a minimal effect, but when we combine with cytokines, now there is a much bigger effect that it enhances the apoptosis. We did the proteomic analysis comparing cytokines and cytokines plus palmitate, and there is a lot of things that change. And one thing that we're very interested is not the like receptor signaling pathway, because this also has also controls apoptosis. And one of the proteins in this pathway is this nicotinamide ribosyl transferase or NMPT. And you can see with fatty acids, just with palmitate by itself, there is no change in abundance. And it is induced here. Oh, sorry, I forgot the legend, but here is with cytokines. It's enhanced by cytokines, but with palmitate is somehow reduced again. Just to localize here, so this enzyme here is from the NAD or NAD metabolism, and is very important for the cells. So for example, the glycolysis need a few NAD molecules, also the TCA cycle, also the red transport chain. So it means that if you don't have NAD in your cell, you basically cannot have energy. There's a few pathways that produce those. So you can make out of tryptophan, or you can make out of niacin. And then there is another pathway here. So NMPT is here in this path called the salvage pathway, because some enzymes will use NAD and then release nicotinamide. And then NMPT is one on the first step that will convert to nicotinamide mononucleotide, and then there is another enzyme that converts back to NAD. These other, the main of the process will not degrade NAD. All these metabolic pathways, they don't, but like process like ADP ribosylation or sirtuins, they do cleave NAD. So what we did was going after NMPT. There is an inhibitor for this, is this FK866. We can see if you treat Th1nk cells with cytokines, and then we add the inhibitor, you have a similar effect, the enhanced apoptosis that we don't see here without cytokine. And then this is reversible if we put nicotinamide ribose, so that comes one step after, I mean, in NumPT, so in the pathway. So it's not NumPT is not required for it. Here is just to show the levels of NAD in the cells. That if you inhibit then with FK866, then you basically deplete the cells, and then you can reestablish with NR. But the interesting part is that NR itself cannot rescue when you treat with palmitate. So if you treat palmitate, you have an enhancement here, but if you put NR, there is no decrease. And you can see at the levels of NAD is also not replenished. So the pathway that I showed was incomplete. They actually need a lot of ATP. So we start to think that maybe there is something else going on that is not NumPT is not the whole story there. We went back to the proteomics data, and then we look at the insulin signaling pathway, because that is one of the pathways that regulates the cell metabolism. And then we see the hexokinase one was regulated in a similar fashion than NumPT, that is increased by cytokines but reduced with the combination of fatty acids. But the response is that if you inhibit hexokinase one with the inhibitor ronidamine, it by itself already enhanced the apoptosis. So we don't think that is actually responsible. We also look at because we also look at the glycolytic pathway and in the TCA cycle doing metabolomics analysis, and then we see several steps that are regulated by this process. It's things like lactate, glutamine, glutamate, malate, and fumarate. And then we end up, the question is like, what happens to the ATP? And then actually ATP is down-regulated by, if we treat with cytokines, there is a higher demand of energy already, so you have a reduction already, but it's further reduced if you add palmitate. So there might be that you don't have enough ATP now to rescue back and produce more NAD. This is again the HPAP data of a single beta cell RNA-seq. We can see that the NAD salvage pathway, you see any NumPT being reduced with one or two autoantibodies, and it means like in this process, we also see the glycolysis being reduced in a similar way in the TCA cycle. So we think this is something like very significant that really happens in vivo. So the summary here is that the free fatty acids increase in both islets and plasma during insulitis. The free fatty acids enhance cytokine-mediated apoptosis in immune sick cells. The cytokine plus free fatty acids downregulate NumPT and deplete cells from NAD, enhancing this apoptosis. And then cytokine plus free fatty acid cause metabolic shift that reduce ATP levels, then further aggravating the NAD depletion. I have a lot of people here to acknowledge. I won't even entertain to go one by one, but it's a really a big collaboration group. It's a lot of coordination between, and these people are really fantastic collaborators. And I thank so much for all the support from the different consortia.
Thank you so much, Ernesto. I wanted to open it up for questions. I see that Camila Ricordi is here from the DRI Miami, and they have the Poseidon trial going on, wrapping up down there. I don't know if he wants to talk to you a little bit about that.
Well, with pleasure. I can say how well. First, congratulations. Sorry, I couldn't join at the very beginning. I missed the beginning, but we've been working a lot on like omega-3 and omega-6 and how you can modulate inflammation and insulin resistance induced by arachidonic acid and high-level omega-6 in the diet. And the Poseidon trial was, in a way, a disappointment because we couldn't complete for two reasons. First, COVID pandemic, that we called the 1 year check of the MTT. But second, because it's such a simple trial done with over-the-counter vitamin D3 and omega-3 that most patients, family, when they see the protocol and the informed consent and say that they may flip a coin and be in the control group, they just go ahead and take on their own the omega-3 and vitamin D. We have spectacular results, anecdotal, on people increasing C-peptide, and now we are publishing, hopefully will be published soon, a direct a linear correlation between the level of vitamin D3 at diagnosis, the 25OH vitamin D, and the severity of type 1 diabetes at onset, measured as a C-peptide that you have at the very beginning. So the more vitamin D, but that is no longer a surprise since high vitamin D level have been associated also with the 14-fold protection against severe COVID or dying from COVID. And you have the VITAL study where omega-3 vitamin D have been associated with a reduction of 39% in all autoimmune diseases. So it's, the study was not completed, but we're just using it. Now there are the recent results from Horvath, and they've been also commented everywhere in the health span community on the combination of omega-3 and vitamin D3 as improving biological age. And I think if there is a correlation between biological age and diabetes and autoimmune diseases on the fact of anything that can modulate inflammation and promote health in a way could help both diabetes, that is an accelerated aging disease, and regular aging on the other side of the coin.
Camille, I have a question for you. Do you see people that are responsive and people that are not responsive to omega-3 fatty acids?
Yes, we do. And I think it would be important to check the non-responders, especially comparing the level of 25OH vitamin D in the non-responders, because the stronger correlation has been vitamin D so far. And then it's like, I don't believe there is a single magic bullet or remedy, but you know, it's an ecosystem that you have to probably implement to protect from abnormal inflammatory and hyperimmune reaction like in autoimmunity. But in a way, it's been shown, there was a JCI paper showing that if you have high omega-3, you double your regulatory T cell level in PBMC, both from non-diabetic and diabetic subjects. But if you have high arachidonic acid inside, your regulatory T cell goes to 0 and the Th17 goes through the roof. So you have a simple like diet-induced background that may be predisposing or make you unable to fight or react to an induction of autoimmunity triggering event. So that in all, I've been discussing also with Jay Skyler and others and Matthias von Herrat on the trial net or intervention studies. I think it would be very important to check the baseline when you start this treatment, or at least retrospectively go check what were the level of AA-EPA ratio or omega-3, omega-6, etc., and vitamin D, especially in those patients at the beginning before you start the intervention.
But
and possibly harmonizing them.
There is some population seems to not be absorbed well, right? That I think might be a probably like something like say gut microbiome, or you know, or
yeah. And the key is like for both vitamin D and omega-3 is sufficient to give a certain dose. Like there are some ridiculous placebo I consider those. Like if you give 1 gram to the population, like the VITAL study said, since Okinawan Japanese people have 1/4 of cardiovascular disease and they eat 1 serving of fish a day, let's give the omega-3 of 1 serving of fish a day, which was actually 0.83 EPA and DHA. But that doesn't make any difference if then you're in America and you go out and eat hamburger, French fries, and fast food as there is no tomorrow, because you have to look at the levels, you should check target level. Are you in range between 1 and 3, between 1.5 and 3 in AAPA ratio, and adjust the dose. The same for vitamin D. At the same dose, there can be a 6-fold difference in the levels that you reach in vitamin D 25OH in the blood. So to me, studies that give just a flat dose of one or the other without checking the actual levels and target that you reach may be less effective in understanding.
I wanted to sort of thank you both very much for those comments and updates. I wanted to just bring that back to the microbiome. Obviously the gut microbiota, contributes to the host's NAD pool, right? Because it makes nicotinic acid, which can be used for for other cells, by other cells for their own NAD synthesis. But can you unpack that a little bit more, Ernesto? You sort of commented a little bit about that in your conversation with Camilo.
Yeah, for the nicotinamide, most of the NAD is produced by the liver. So in other tissues, the main pathway is actually the salvage pathway. So if you mess up with the salvage pathway, then you're gonna end up having less than you need of NAD molecules.
Could the, if you have the salvage pathway somehow affected and messed up, could the microbiome come in sort of as a backstop, as a rescue? Is there enough, are they producing enough or no? Or enough of the precursors?
To be honest, I don't know exactly the numbers, but some of these pathways, like sirtuins, they use a lot of those. And NAD is present, I think, in millimolar in the cells.
Yeah.
So it's not as easy to, it's just like uptaking it to compensate that.
Yeah. It's not really a backup.
Yeah.
Even though, it is kind of strange too, at diagnosis, point of diagnosis of type 1 diabetes, the microbiome undergoes an alteration and it's, it becomes less diverse and there are changes. So it's kind of strange. Could be anecdotal. I had a question for you also about, the recent Matt Marins and Richard Banniger paper. They recently published a great preprint in August of 2024, GK activity control subpopulations of beta cells that alternatively lead to islet calcium oscillations. Could GK in these leader cells be more susceptible to alterations in fatty acid balance? And could the leader cells going offline first, set a cascade up?
So what happens is that when you have too much fatty acid, then you shift your metabolism from towards more TCA cycle than glycolysis. So there is that balance, right? You put more fatty acid, and then for you to use that as energy, you need to do more respiration than glycolysis. So that is part of what I think is happening.
Very interesting. And then we talked a little offline, Kyle Galton, Mikey Sander, and Rebecca Melton, just out of UCSD, dropped a really great preprint today in bioarchives with some really fantastic tools dissecting, full islets, really looking. And they saw an enrichment in some of these 29 T1D donors that they looked at in steroid hormone receptors, stress responses, but they also saw decreased activity in HNF1. So I wonder, it might be kind of interesting to use some of those tools they're developing to monitor some of these specific lipid changes that happen in the islet, subpopulations. It'd be very cool, I think.
Yeah. I haven't read the paper, so I haven't wrapped up my mind yet. But as you can see, like we get all the data what other people publish as well, and we try to integrate to to understand what the whole biology behind it. So
Yeah, you guys
any
are
other
great.
data is welcome.
Yeah, you guys are doing some great collaborative work. Really, hats off. Any more questions, anyone? Karsten? He's here, and some other
Oh,
there.
you muted, Karsten.
Oh, I'm not muted. Somebody's trying to unmute.
No, I think Karsten was trying to say something, but he's muted. Yeah.
Oh,
I think you can hear me now. I came a bit late, but it's highly interesting, and there's been a trial on nicotinamide and in human pre-diabetes or early diabetes, and one could try to add, if that should be repeated, these omega-3 acids and whatever you have mentioned. That could be because it was a surprise that it didn't work at all at that time.
Yeah. Yeah, because I think based on our data, just nicotinamide might not be enough. If you have because we couldn't get back to NAD, right?
Yeah, that was the problem. But maybe one could add some of these things or omit some of the palmitate or whatever it is.
Yeah, because the omega-3 fatty acids had the opposite response. Maybe if we combine then nicotinamide with omega-3, it might be a way to go.
And maybe you omit some of the fatty acids you mentioned. Yeah, but highly interesting.
Thank you.
Yeah, it's great. It's great that you guys
I'm a strong proponent of combinations strategies for forever, even in islet transplantation, but it's very difficult to get it past an NIH study session because is add the complexity. But we are using, just to let you know, as a follow-up to the Poseidon, we are now using vitamin D, omega, the APA, DHA, but also a combination of polyphenol sirtuin activator that include pterostilbene, polydactin, onychiol, and ellagic acid. And the surprise that came from the initial studies was we were doing it to increase C-peptide, but we noticed a regression of retinopathy in some cases that is completely to be determined what could be the mechanism. But sometimes you learn from combination strategy, and then you have to go back to the mechanistic study and try to understand what the hell you're doing. But at least they are not biologic with side effect.
Great. We'll have to reach out to the DRI and get some more and have a talk with you guys once you get that out in a publication. It's really, really exciting.
It's all in this site. I write it in the thing. Since I don't have any more time giving my age to do long prospective phase 3 trial.
We'll have to get a trainee then.
I put everything in a website, but let me see. Wait, I send it only to Ernesto.
Great. All right. Well, hopefully you all will continue the conversation. Thank you so much, Ernesto. That was really exciting work and interesting collaborations among many different scientists to pull this off. Appreciate your time and look forward to what you come up with next. And thank you very much, Camilla, for adding that link in there for everyone to take home. I hope you all have a great rest of the day, and we'll talk again soon. Thanks so much.
Thank you.
Bye-bye.
Bye.
Thank you.